Hi, everybody. This is the review recording. So basically, you'll just see my little on-screen thing, as many of you recall. And I'm just basically going to start to do the recorded review for exam one. Again, please, if you want to throw this thing and just load it up into some AI app and figure out let them make a test for you, it works. I have no objection. As far as I'm concerned, that's a perfectly usable tool. I know that there's others, in education who have a certain degree of objections to this, but to me, it's anything that helps you study and get answers correctly is a useful tool. So let us begin on that note. And this exam is, I'll give you I think we got 65 questions that are the usual multiple choice, true, false, matching type stuff. And effectively, that's what it is. And it basically, I think the major point of emphasis is the chapter that effectively that chapter 3, the gram negative, gram positive structures, peptidoglycan, endospores, the endosymbiotic theory, and materials like that. So let's move on from there. So with regard, first of all, I like the historical figures. I think they're important to understand what goes on with microbiology. The big deal is Pasteur usually basically advocating what was called germ theory and to oppose the theory of spontaneous generation or abiogenesis. That's because finally we had a historic figure, a noted scientist and biologist who had accomplished a lot with regard to the wine industry and had been able to stop wine spoilage by something we now call pasteurization, which we're going to be studying more specifically. You don't At this point, you don't have to know the numbers for pasteurization or anything like that. That's actually in the next chapter we'll start to cover next week uh, with regard to methods of control. And he was able to advocate a theory and then prove the theory, or at least more or less prove the theory. Certainly had the right idea with regard to the theory of things that we could not see that were visible microscopically. And he called them germs, and we still use the term today, obviously. And the reality was he was fighting this notion of spontaneous generation, that there was some force in the air, that some life force that could create things, and it was so contrary, and it was really the advent of modern. If you want to think of the advent of modern science starting in the Renaissance and carrying it over, and this was in it, and changes then took years and years and years. It's not It's not. It's so different today when all of a sudden molecular, are being able to use the techniques has changed dramatically in something more than 50 years that I've been around, and all the techniques, the technology, et cetera, et cetera. And people are more willing to because they're looking for answers. So it's very accepted. So on the other hand, the other really great figure in this was Koch. Again, were rivals in the sense that Pasteur was French and Koch was German. And while Pasteur, he was all about helping people, he really developed the first treatment for rabies. Uh, he was able, so he basically was looking for treatments for various things. He was a problem solver with a lot of credibility, as opposed to Koch, who, not that he felt that you needed to study things, and he was really an advocate of something you're doing in the lab, which is growing things in pure culture. He developed something called Koch's postulates, which we'll look upon in a subsequent chapter. And he basically developed the lab techniques we still use today. His the scientists who worked with him developed agar, developed ways of isolating things. He was identified anthrax, and that was the foundation of Koch's postulates, because it was destroying farm animals and also people as well. And we'll go into how that works subsequently. And so whether it was staphylococcus, streptococcus, tuberculosis, Bacillus anthracis, all of the he he identified and grew in pure culture every possible bacteria that he came in contact with, other than the ones that he did not know that they could not be grown in pure culture. So it's really significant that these two, again, had we're constantly sort of outdoing each other with regard, but to the betterment of mankind. And there were other historical figures, certainly. We had, you might remember me talking at length, but even before we looked at, because I tried to do it historically, and whether it was Jenner with the smallpox vaccine and the cowpox, basically isolating something from cowpox, taking a gigantic risk to develop, basically a way of preventing smallpox that became so successful as time as things refined and matured that we effectively eradicated disease in a 200-year span that was one of the greatest killers of all time. And how Semmelweis, and this was really, to me, it always underscored how doctors can't say bad things about other doctors. You're sort of, and it's easy to get ostracized. Doctors have big egos, as many of you will find out. Um, Basically, he was the one who looked at the relationship with hygiene and soil material, developing something called puerperal sepsis or childbirth fever, which is streptococcal, and that really gave him the clue. And seeing how the midwives didn't had very, very few cases, and doctors coming up to deliver babies from the lab, which is really basically they were just was mortuary. They were just dissecting things and trying to figure out what killed people and understanding the anatomical and physiological processes, and didn't barely wash their hands, had old soiled rubber aprons, and yuck. So, and when he wrote that article, even though it worked brilliantly in Vienna, they ostracized him. And I mean, it's just because he basically said, "Hey, oh, well, doctors can't be hurting people." In this case, they didn't know any better. But Lister took that information and took Pasteur's information and really became the father of modern aseptic technique. And so we began to utilize dilutions of something, again, we'll look at in the next chapter, called phenol. And it really was sort of just a special ingredient in a lot of uh, antibacterial soaps until just recently when they found something that was better and less problematic. So he really was in surgical techniques, washing your hands, cleansing the patient, cleansing the instruments, and it's exactly what we do today. And obviously it's been refined over time. Again, I made a big point about genetic engineering, the magical things that bacteria have given us for good, and much more for good than for bad, even though we inevitably will focus on the bad. Okay, it's a textathon today. It's my daughter leaving for her first anniversary trip. So I apologize for the interruption, but I can live with that one. If you hear a ding, it's inevitably people contacting me. Nevertheless, so we talked about genetic engineering. They're using little fragments of DNA and basically splicing it onto other DNA. We'll look at the process again in the next major unit, the next test unit that we do, when we talk about something called conjugation. And basically, that's genetic material, DNA, and we utilize E. coli and yeast principally for the thing. I mean, there are other techniques that are available, but for our purposes, it's DNA with E. coli and yeast. Don't forget the distinction between resurgent and emergent diseases. Emergent diseases are mostly mutations, but we're going into new areas and diseases we have not previously identified. So we call them, and they are emerging. So technically, so whether it's COVID-19, which is effectively a kind of mutation, whether it's MRSA, which is a kind of mutation, whether it is these different flu strains or different strains of RSV that we'll talk about down the road, all of these are emergent diseases. And again, it's mutations, and we'll explore that. When we talk about resurgent, these are diseases like tuberculosis, like measles, that have existed for a long, malaria to a certain extent, that have existed for a long period of time. And and there's really four major reasons that have caused us to encounter them again. With measles, vaccine laxity, worldwide travel, people living longer, and then the whole idea, the whole giant industry now of immunosuppressant therapies and immunosuppressant diseases. Okay, it started with HIV, and then we began to look at medications that would address the immune system, and tuberculosis became a significant problem. Down the road, you learned it really defines AIDS in certain cases. But The reality is that now we what we call biologicals, and these are genetically engineered treatments and medications that are highly expensive. And what they do is they suppress your immune system. If you have rheumatoid arthritis, if you have psoriasis, if you have systemic lupus erythematosus, if you have certain kinds of immunological activities, all of these things potentially are fraught with danger. It's a two-edged sword. And so that's really, and it's not something we're getting more people that are immunosuppressed. Uh, it's not. I don't know how well it's been studied. There is it environmental? Could be. There's a lot of things that we're seeing. Different diseases than we did years ago, and a lot of it has to do with technology and perhaps alterations in the environment, and a lot of things. And people often implicate genetically modified things in our food source, and with our consumables, which is not my area of expertise. I'm not trying to start telling you not to use this stuff or anything like this. It's hard to avoid. So I think you really have to look at all those things. And I went and in the introductory stuff when I was giving you like what differs bacteria from archaea and viruses and things, those are all things you need to know. You can't get the question wrong. When I say unique, it means nothing else have it. That's peptidoglycan in bacteria. Okay. Algae having chlorophyll. Those are all significant things. So the distinction, when things are similar, they're more subtle distinctions. Bacteria and archaea, single celled, they have cell walls, but bacteria, peptidoglycan, and archaea, okay, have do not, but they live in extreme environments which really defines them because bacteria can exist there, but more likely they're not necessarily always found there. But they're very similar in size, and they're very similar in other ways in their in their internal makeup. So that's why they're categorized in the domain system differently, because then you're getting down to molecular aspects of their RNA and their DNA. But in general, if you were to show an illustration of each, they would pretty much look identical until you really explored the molecular aspects of it. Just so that's something to keep in mind. And again, what are viruses all about? Viruses, as you will learn, can infect any living thing that causes archaea, eukarya, bacteria. It doesn't matter. Us, we all know. So viruses don't necessarily play favorite, but again, they contain, and you'll learn a lot about the fact that they contain neither. They have to contain living things have DNA and RNA. Viruses, it's DNA or RNA, never both. I'll say that 1,000 times. Probably have already. And the big distinction, because we call them acellular forms with those and prions, is that prions are proton only. Proteins, I beg your pardon, only, and they are misfolded proteins that effectively bang into other proteins and deform them as well. And so it's again, these are kinds of things we explore more as we get into those chapters. This is the introductory nuggets. And so now we get more into the bacterial aspect of things. So one of the areas I touched on were endospores. They effectively live hundreds and thousands of years. They are in the what we call the endospore stage. The endospore stage or the spore stage is a state of suspended animation. It's a fight against extreme environments, as we will go on and talk more about. So the endospore is something, and the distinction went back to, again, and this is a big distinction one, but when we talk about something in a vegetative stage, okay, bacteria that are growing and healthy are in a vegetative stage because we used to think they were plants. So they go between the ones that can, and it's typically the species of Bacillus or Clostridia for our purposes that are able to undergo these transformations, which leaves, all the others really don't. Most of the bacteria otherwise they're either in a vegetative stage, and if they can't be growing like E. coli or staph, then they die. However, these, when conditions are adverse, can change into the stage that basically, as I say, is a state of suspended animation, a dormant stage, whatever word you want to use. They have, remember, they asymmetrically divide. What is going to become the endospore, we call the forespore. It's surrounded by the mother cell. There's peptidoglycan forms between the two membranes. The coat is made out of proteins. There's additional protein layers. So we have the cortex, and then which is really the coat of the spore made of proteins. Those are all significant features of endospores. And yes, when conditions are no longer adverse, and what does adversity mean to a bacteria or to these bacteria? Not enough nutrients, carbon or nitrogen containing nutrients, and or not enough water, because water is essential for all living things. And then you learned about this in lab and things like that, the different forms, the distinction between cocci and bacilli, you know about that. In eukarya, you're familiar with phospholipid bilayers because they're in bacteria too. So the phospholipid bilayer is a typical membrane, except bacteria have a cell wall. And then the big distinctions we'll get to is the gram-positive and the gram-negative cell wall. So again, living membranes, and they're all living membranes, whether they're inside microbes or on the exterior of microbes. Phospholipid bilayer, embedded protein, semi-permeable. That's really what they are. And it's all about what can get in and what can get out. And so I went into all of those in extraordinary detail that are there. Again, the big distinctions between our cells, if you want to look at it cellularly speaking, the DNA in bacteria and archaea is a big circle. Yes, it twists and twists and coils, but it's effectively a big circle. It's not linear. We have linear DNA in pairs in eukaryotic cells. Bacteria replicate by binary fission. Eukarya typically replicate by mitosis or meiosis. Yes, there's budding and things like that yeast do. Is a little more simplistic process, but effectively even the budding is a kind of mitosis. When you actually look at it, in close, which is not something we typically do in this class, that are there. And the other big distinction is the ribosomes. Eukarya have 80S ribosomes. Bacteria have 70S ribosomes. The only place eukarya have a circle of DNA or an 80S ribosome is in the mitochondria, and that's the whole basis of endosymbiotic theory. This structure was probably an ancient ancestor of a bacteria that got swallowed up by this common ancestor cell, and magically, rather than destroy it, incorporated it and became effectively an energy factory. That's why we can make more mitochondria when we have increased activity, and they'll disappear since they're no more being made, and they have a lifespan. It's not like they're forever structures, and effectively that's because they have their own DNA and their own RNA, except it's bacterial DNA and RNA. Again, the circular chromosome, the single circular chromosome, and the 70S ribosomes. That's so important. The structure of peptidoglycan is important. Peptidoglycan remember, I know it's complicated. This is a learn once and throw it away thing, but you should be familiar with it, because I'm always worried about things, particularly for students taking licensure exams like the NCLEX. It's going to come around and bite you, and this is what does it. So we all teach it. Okay, it's only in bacteria. That's what makes it unique. Those alternating NAMs and NAG units. The facts that it's stapled together. Remember, the glycan is the NAM and NAGs. The bricks, the mortar is the tetrapeptides and the interpeptide bridges, which are only in gram positive. By the way, that's not a big deal. The big deal is it's in different amounts in different bacteria. Gram negatives have a thin layer or two, and there's anywhere between 20 to 40 layers in gram positives. And again, those NAMs and NAGs, okay, the big N, okay, I remember it this way, it means it has nitrogen. Nitrogenated carbohydrates are biological structural materials. That's what's in our connective tissue. It's not a whole lot different when you think of the generalized structure of bacteria as well. And again, the other thing that's important about this is the distinction, really, in the gram negative, that whole unique lipopolysaccharide layer. Again, another word, unique, because it's only in gram negatives. And the lipid A portion is always a toxin. The O antigen portion is an identifier. It's associated with different things, but in and of itself, it's not a toxin. It just helps us type or identify specific toxic forms that we have sort of a history of. We have a book on. Oh, it's that one. Oh, it's this one. Just the same way we have different kinds of staphylococcus and streptococcus, so we know which is which. E. coli it doesn't have as many species, but it has these different things, and that's when you'll hear O157 and H7, and the way those markers, the O is on that LPS layer, that O antigen, which is external, pointing out, and the H happens to be on flagella, but I'm not going to ask you about that. And so it's associated with the gram-negative outer membrane, and it's only that outer leaflet. Other than that, the inner membrane is a phospholipid bilayer. The inner leaflet of the outer membrane is phospholipid, but it's not a bilayer because they're different. And that's really very, very significant. Remember, both gram-positives and gram-negatives secrete toxins to do damage frequently, but that lipid A layer is the unique, we call that the endotoxin. Exotoxins come from both. The endotoxin must be gram-negative. The question says, this is a problem with an endotoxin. The answer better be you go search for a gram-negative, like E. coli, like Proteus. It doesn't matter, like Neisseria. That's what you search for when we go farther along with tests and you see this evidence or you see this wording later on in your careers. Oh, forgot. Again, teichoic acids in the gram-positive cells, that very, very thick layer, that's how they sort of attach it or adhere it. And so that's a feature that gram-negatives don't have. That's how the peptidoglycan. Here, peptidoglycan's porous, so gram-positive cells don't need any pores or porin proteins or transport channels or anything, transport proteins, because they're effectively porous. But gram-negatives need them in both the outer and inner membrane for water and for things dissolved in water to be able to get in and out of the cell. Gram-positives don't need that because in the outer membrane, because they don't have an outer membrane A, and the peptidoglycan is porous, so that's important. But all of them have that sugar shell, that glycocalyx. And what happens, it's made of polysaccharides, that's what it means. Okay, lots and lots of sugars. And they can be, if they're not well organized, they're very sticky and they tend to become a slime layer. If they're very, very thick and organized and they might have certain different materials associated with them, then they become a capsule. And again, capsules have a great deal of significance with regard to disease because they impede and many times completely inhibit or prevent phagocytosis. And again, that's our body's big aspect of our body's defense. We identify things, gobble them up, and destroy them. So it's really important. And then I frame a lot of questions. They'll either say prokaryote or eukaryote. Identify that first. Okay. Sometimes they're highlighted. Sometimes I'll say in bacteria or in prokaryote. I've tried to go around and highlight them so you don't have to. But I miss them, and it's not by intent. Okay. Because when you have a lot of questions and a lot of words, you tend to miss things that you might want to highlight to help you guys. So understand the prokaryotic. So, for instance, the 9+2 pattern is something of microtubules, which we don't even have in bacteria. Bacteria don't have microtubules. They don't have intermediate filaments. Okay, they're only in eukaryote. They both have actin filaments, or what are often called microfilaments. Of those, what we call those CAMs, those cell adhesion molecules, those are the only ones that are really shared by both the actin or microfilaments, because they can, remember, they can assemble or disassemble. It's an A and P thing. The intermediate filaments are insoluble. They basically create that. Remember, eukaryotic cells have that very elaborate internal structure and that interesting 9+2 formation of microtubules that we see in flagella and cilia. In prokaryotic flagella, it's different. First of all, in eukaryotic cells, they're internal appendages. They just, remember, they push out the membrane and it sheaths around them. So it looks like they're sticking out, but they're not. As opposed to prokaryotic cells, where actually the protein appendages like flagella, the longest, cilia, I beg your pardon, there are no cilia, pili, which are shorter, and fimbriae, which are shorter still. And the fimbriae are for mostly attachment. And the pili, you're going to see, can be attachment, but they play a role in genetic transfer mechanisms. That's why sometimes they're called sex pili, but really they're just a hollow protein appendage. The flagella are made of flagellin, a protein that protrudes, just a chain. The pili are made of pilin, okay, a protein that protrudes, okay. And they really don't identify a specific protein that is probably similar to both of those that make the fimbriae. And that's their size thing. And remember, eukaryotic cells are much, much larger. So a flagella in a sperm cell is enormous compared to a flagella in a bacteria that's protruding from a bacteria like E. coli. And so the big one that, and we're going to get into in depth, we call it the F pilus or F pilus, and which F is for fertility. Even then, they thought it was sexual, but it's not. Bacteria are completely asexual. That's the one that's made out of, okay, that's the one that's made out of this pilin. It's hollow, and it's for transferring genetic material, DNA, and sometimes we nickname it or erroneously call it a sex pilus or a sex pilus. Again, that's really important stuff. It's there. Plasmids, again, single circles of DNA that are by definition non-essential, meaning they can come and go. Bacteria don't have to have them to survive, but they are useful, and principally for transferring traits from one bacteria to another that enhance their ability to survive and thrive, particularly antibiotic resistance. Okay, and they replicate right along with their DNA. I mean, the nucleoid is where that big circle of DNA, really what that is, that all that compacted DNA, supercoiled, that big circle is called the nucleoid. That obviously replicates. This is a small circle. That obviously replicates. DNA replicates at every cycle. It doesn't matter whether it's all attached to one another or not. So I think all of those things are important. Let's see what else. Oh, just remember that whole look at the animation on endospore formation. Okay, remember, you have the forespore and the mother cell, just to review it. Remember, you have peptidoglycan. You have limited nutrients. That what starts it is the adverse condition, limited nutrients and or limited water supply. Okay, it takes hours to do. Okay, and effectively, that the role of that dipicolinic acid, that interesting chemical, what it does is it attracts calcium and it spits out water, and that really creates that environment. It's almost like a kind of dehydration. It's not, because there's a limited amount of cytoplasm, which is a water base inside the endospore. But I'm trying to use something creative way of describing it so it sort of cements itself in your mind. When we talk about eukaryotic cells, and we did this before, all eukaryotic cells have sterols. The only bacteria that utilizes a sterol that we study is that mycoplasma, because it's the only one that does not have a well-formed cell. Not the only one, it's the only one that we talk about that does not have a well-formed cell wall. Okay, but we utilize cholesterol. Remember, when we get to fungi, I talked about ergosterol. Sterols just mean they come from the cholesterol nucleus, but that something has to stabilize the cell membrane. For our cells, we couldn't survive without it. Okay. And again, a much more elaborate cytoskeleton internal structure in eukarya v prokarya. Don't forget, when we talk about eukaryotic cells, the interesting thing about the mitochondria and the chloroplasts, which you would find in algae, and mitochondria you would find in all eukaryotic cells, is effectively they're about energy transformations. So we have this internal energy creating structure. It's very much different. Okay, we don't have membrane-bound organelles in prokarya. The energy has to be done along the cell membrane. There's all sorts of stuff that's associated with it, but they're only found in eukarya. Nothing. You might have isolated bits of chlorophyll in exotic bacteria, but you will not find chloroplasts, or you will not find mitochondria, because they're membrane-bound organelles. They're only in eukarya, and they're all about energy transformations. So that's really important as well. Oh, and I would remember, take a look at that gram-negative cell and look at it from going outside in or inside to out, the layers. Remember, you have the outer membrane, because I want to make an emphasis point about this. And you have the inner membrane. Between them is the peptidoglycan layer or two. And that's contained within the periplasm. And then you get to the interior of the cell. So remember that. Okay, you have that outer membrane or lipopolysaccharide containing membrane. You have periplasm. You have the phospholipid bilayer membrane, and then you get to cytoplasm. So because you've got to know the sequence to identify things. And other little nuggets about those. My dog's giving me a hard time. I might have to stop, come back and do this. He's being a pest. He's not barking. Uh, we talked about different categories. I forgot to mention that mold, yeast, fungi, they're saprobes or decomposers. They live off of dead stuff. And I'm trying to get through all the bacteria stuff. Okay, hold on, hold on. Hold on, hold on. I'm getting to the stuff we have recently just talked about. A couple of other things. I'll do that. I'm almost there. Oh, two words. And I really got the word facultative. We talked about a little bit with facultative. Facultative bacteria grow anywhere. E. coli staph. Facultative means they have the ability to grow anywhere. An important word for bacteria that require a lot of TLC and a lot of extra nutrients is called fastidious. I hope I mentioned it in class. I'm going to try to remember to do so tomorrow. Obviously, I'm doing this on Thursday morning, the 12th. This 13th will be our next class. Fastidious needs a lot of growth factors. Big important difference. It's a lab term as well. Fastidious versus facultative. So if you know somebody who needs a lot of attention, and you got to constantly do stuff for them, they're fastidious. And if somebody's low, it's sort of like low maintenance versus high maintenance. If that's still the parlance of today, I think that run, tumble, run. Don't forget that. Okay, remember it's all about the more flagella they have. The different is it peritrichous and monotrichous means one. Peri means all around. They move the most. And they can be at both ends. And remember what attracts them. They could be mostly it's chemical, but it can be light, it can be air, it can be magnet. All of those types of things can attract them. There's so many different things. Heat, there are different bacteria that have to go away from warmer areas or have to go toward warmer areas when we looked at those categories that are so important. So I think that's important to understand. Again, they're strictly asexual. I just want to finish the bacteria before I go into that last chapter with how they grow that are there. Covered. I think we got most of it done there so far. I just have that last stuff that I'm hoping the dog lets me do this. No guarantees he will do this. Okay. Okay. So everything now is growth. I apologize for that little in between thing, and hopefully the dog will let me do the rest of this without interruption. But I'm not guaranteeing it because he's like that. So if you hear me stop, I'll have to get to the rest of it. There. Okay. So now we basically are getting to these different categories. First, the heat category. This is how they grow. Remember CHONPS for the six things. That's carbon, hydrogen, oxygen, nitrogen, like both eukarya and prokarya. But the phosphorus and sulfur, the P and the S, are the big distinction between. It's usually calcium and phosphate and eukarya, in us particularly. But phosphate and sulfur are very, very specific to bacteria without going into all the other forms, which I don't recall off the top of my head. Okay. So the temperature ones that are important, again, the three. Only one I'm asking about. The hyper ones are extremes, and they're effectively archaea. Mesophiles, most of the disease-causing bacteria are certainly in that category, particularly that affect us, because they flourish at the same temperature that we do. They're in that 25 to 45 range. The ones that are those psychrotrophs, psychro with an R, trophs, are the ones you find in the fridge. They're typically spoilage organisms, so they impact us. And then the thermophiles, the ones that they probably go from like 40 to 70, that kind of an area within degrees Celsius, even though it's not, we don't encounter them as much, but certainly there are warmer areas. And I made a big point because it's really significant, the whole idea of hot tubs and those kinds of environments, areas where we go to heat, think of a sauna and things like that. That's why, keeping them in a, disinfected and maintenance of those things. And particularly, a lot of us go to the gym and use health clubs all the time. It's very, very popular now. I think you have to be familiar. So those are the big three I think you have to be familiar with, more so than anything else. And then the oxygen levels. If you are an obligate aerobe means you have to have mechanisms to deal with those toxic derivatives of oxygen. So you need both superoxide dismutase and catalase. You got to remember that because they take care of it. If you're if you're an obligated anaerobe, you have to, okay, form in the absence of oxygen. Oxygen is destructive. Oxygen is immediately toxic. You have no means of dealing with it. Okay. Okay. And then everything else is in between. If it's a facultative, and really a better term is facultative anaerobes, I know the Cowan text calls them, and they really are not. They are typically facultative organisms, are or function well in aerobic environments and ferment in anaerobic environments. So, I mean, I would probably, I think I would adjust the terminology for our test purposes. We call them facultative anaerobes slash aerobes. The meaning facultative means they grow easily, whether they're growing in the air or not. So they can grow in the air and they can grow without the air because they can ferment. But probably a better way to describe it. Okay. But a lack of oxygen is not going to be fatal to them, is maybe the best way to describe it. Okay. Let's see. So again, you need to know those, kind of know those temperature levels. I think that's important. 25 to 40, the 40 the 40 to 70, the one for the whichever one there is, and I'm not seeing it off the top of my head. For the cycrotrophs, where they go, I think it's 5 to 15, but really it overlaps a little bit more. That's the spoilage organisms. Okay. So again, really, why we refrigerate things, it's slowing down things. When we chill things, the metabolic rate slows. When we freeze things, effectively, temporarily, it stops. Okay, let's see. Oh, the microaerophilic bacteria, the little ones. They just can't handle large amounts of oxygen. That we find them in areas like in tissue, devitalized tissue. That's that one associated with flesh-eating disease. So tissue that's been damaged, or like in diabetics that have bad circulation and proportionally are getting insufficient oxygen. And again, the one that involved in gastrointestinal ulcers or gastrointestinal cancers, Helicobacter pylori. It is again, it's by definition one of these microaerophilic structures. Interestingly, it burrows into the stomach, but we don't need to get into all that. We talk about that a little bit. You may talk about the AP2 and GI when you get there, but they have limited means. So I think that's important to remember. And remember, when we talk about toxic derivatives of oxygen, the whole ball game for this problems with the air, they occur normally. Anytime you use oxygen, they're going to occur. Okay. And yes, they're the foundation for cancer theory. They're the foundation for cancer theory, for aging theory, for a lot of things that start degenerative process and deteriorating process in tissue. They're there. And we have, within the peroxisomes, we contain both catalase R, specifically in eukaryotic cells, and peroxisomes have catalase and superoxide dismutase. Bacteria don't have peroxisomes because they don't have membrane-bound organelles, but they make some of it internally. And again, that most of the bacteria we do are classified as neutrophiles, and they function. We typically see them between pH 6 and 8. Even though we're 7.35 to 7.45, bacteria have mechanisms. And again, that for them, 6 to 8 is relatively neutral. They can handle a little more acidity and a little more alkalinity. I think the cell wall probably plays a big role in that. And remember what happens with regard to osmosis in the environment, the tonicity. Isotonic, well, since it's the same solute to solvent concentration in all living things, will have no impact. Okay, so normal saline, whether it's a eukaryotic or prokaryotic cell, won't adversely impact it. Hypotonic, pure water, the cell wall of the bacteria will prevent problems. Unfortunately, it'll cause eukaryotic cells to burst if they do not have a cell wall. So a plant won't be a problem, but a protozoan would be a problem. Okay, an algae would not be a problem. And then lastly, the real one that affects both of them and why it is so useful is hypertonic. That's salting. So salt water or brining things, that's preserving because it doesn't let, particularly with foods, it doesn't let bacteria grow. We do that the acidity is a big nugget too, because when we pickle things, we're preserving them. Yes, it denatures the protein, but it preserves them. So pickling something means putting something in acid. And we'll get into preservation and food preservation a little bit as well. My favorite fun area. So what happens in a highly salted environment with bacteria? Plasmolysis. It pulls the membrane away from the cell wall. Remember, it's an envelope and it bursts. Okay. And again, autotrophs, photosynthesis, they utilize carbon from carbon dioxide gas. We call that carbon fixation. Okay. Heterotrophs, just like you and I, they eat basically everything, and that's where they derive energy and building blocks. There. We did not do the bacterial growth curve. There are a series of 6 questions on it. It is super important. Okay. There are 4 phases. I'll give you the answer key. A, lag phase, B, log phase, C, stationary phase, D, death phase. You know to know the events that occur in each one of these. You need to know the difference between what are called primary and secondary metabolites. For instance, lag phase, when you and I, just like when you got here to school and you're as an incoming freshman, or you're in any new environment, you start a new job, that's lag phase. You've got to kind of get used to things. And we don't see growth, we don't see changes. So bacteria, they become acquainted with their environment, they start to feed on the environment. It takes a while to ramp up and do reproduction. Log phase is called exponential growth. We're going to go over the growth curve at length. That's tomorrow's exercise. They grow extraordinarily rapidly. Understand that is the best time to attack them. They grow so rapidly, if you put antibiotics or any agent in to destroy them, they will gobble it up just like a hungry animal and they will be destroyed. In a closed environment, like a test tube, or particularly like a culture plate, what will happen is they enter what's called stationary phase. It's a gradual transition. You got to push the button for the coffee. I have it loaded. Just have to click it on. Yeah, I'm doing that. And so in stationary phase, so what happens, there's a transition. Stationary phase means it's entering a period of time where there's growth is adverse and there's not enough nutrients, so endospores can form. It's also when they try to defend themselves, that's when antibiotics form. And then finally, when nutrients disappear completely and there's nothing else to feed on, they enter something called death phase. So I'll go over this at length, but that's the short course of it. And we're almost done. So I think I've covered everything else that's there. It appears that I have. Okay. Oh, yeah. And lastly, with the growth curve, and you're going to see E. coli, why we use them so much. They're so easy to maintain. And as long as the conditions are favorable and they have what they need, some salts, some glucose and water, and they will replicate every 20 to 30 minutes unstoppably forever. That's why I use them so much in research. So thank you. I will send this to all of you as soon as I convert it to the YouTube. and ask me any questions, particularly as over we have a whole week now plus to study for this. Thank you. Bye bye.